Hello and welcome to lecture 15 of our course on stochastic modeling of biological processes. In sections 4.1, 4.2 and 4.3, we studied diffusion in containers with reflective boundary conditions. In section 4.4, we will consider that the boundary or a part of it is fully adsorbing. That is, whenever a molecule hits the boundary, it is removed from the system. Typical questions that we would like to answer include: what is the probability that the molecule is captured by the adsorbing boundary? What is the average time for a molecule to be adsorbed? If we keep the density of molecules constant far from the boundary, what is the average rate of adsorption? That is, how many molecules are adsorbed per unit of time. The answers to these questions will depend on the geometry of the domain. We will start by discussing one-dimensional problems. Later, we will also discuss problems in 2 and 3 spatial dimensions. First, we will consider diffusion of molecules in a one-dimensional interval 0L, with a fully adsorbing left boundary at x is equal 0. We will assume that the right boundary at x is equal L is reflecting. Then the time evolution of the deterministic model will be given by the diffusion equation for density pxt of molecules at point x at time t. The left boundary is adsorbing, so we have the boundary condition p is equal 0 at x is equal 0, and the right boundary is reflective, so we will have boundary condition derivative of p with respect of x is equal to 0 at x is equal to L. In this video, we present the simulation of 2000 molecules, which are initially uniformly distributed in the interval 0L. So our initial condition for P is the constant function, equal to 2 molecules per micrometer or 2000 molecules per millimeter. The red line is the solution of the diffusion equation, while the gray histogram is calculated by the SDE-based model of diffusion, which simulates the trajectories of individual molecules. We observe that molecules are removed at the left boundary from the system, which results in lower concentration of molecules at the left boundary as the time progresses. The final snapshot of this video is presented as figure 46b in the lecture notes. It was calculated using the SDE-based model of diffusion. In figure 46c, we present an implementation of this model using the compartment-based approach. At the level of spatial histograms, these models provide equivalent results. To calculate these results, we can reuse most of the computer codes from sections 4.1 and 4.2. We only need to add the implementation of the adsorbing boundary at x is equal 0. Considering the compartment-based model, we divided the computational domain interval 0L into k compartments of length h is equal L over k. We model the diffusion process by the following chain of chemical reactions, where A with subscript J denotes the number of molecules of the chemical species A in the jth compartment, and the jump rate between compartments is given as the diffusion constant divided by h square. To model the adsorbing boundary, we add another reaction to the model. It is a jump from the first compartment to the left, where the molecules are removed from the solution. This extra reaction is the only modification of the model. In particular, we can again use the Gillespie algorithm to simulate the chain of chemical reactions for 13, together with this extra reaction for 74. Considering the SDE-based model of diffusion, we simulate individual molecules. In our illustrative one-dimensional example, the position of the diffusing particles evolve according to equation 462. The resulting stochastic simulation algorithm is given as the SSA A13E13 in the lecture notes on page 123. To implement the adsorbing boundary condition at x is equal 0, we add the step C13. If the position at time t plus delta t is negative, then the particle left the simulated domain through the left boundary and we terminate the simulated trajectory. While this might look like a reasonable approach to implement the adsorbing boundary condition, in the SSA A13E13 we include an extra step, D13. If the position at time t plus delta t is inside the simulated domain, we terminate the trajectory with this probability. This is explained in figure 4.5a, where the time t is on the vertical axis. This is the position at time t and this is the position at time t plus delta t, calculated by equation 462. In this simulation, you can see that both positions at time t and time t plus delta t are inside the simulated domain. However, equation 462 provides a good description of the underlying stochastic differential equation only if delta t is sufficiently small. If we decrease the time step, the particle will not go straight between these two positions, but it will follow a stochastic trajectory. 5 realizations are visualized here using different colors. You can see that the red realization and the green realization cross the boundary x is equal 0. So these trajectories would be terminated if we used a smaller time step, while the other trajectories stayed in the domain. The probability that the trajectory crossed the boundary is given as formula 363. Its proof is left to you as exercise 4.8. Therefore, in step D13, we terminate the trajectory with this probability. As expected, this probability is close to 1 if at least one of the positions at time t and time t plus delta t is close to the boundary, and this probability approaches 0 if both positions at time t and t plus delta t are far away from the boundary. Our next question in this section is: what is the average time for a molecule to be adsorbed? To answer this question, we can apply our result from chapter 3. Let tau y be the average time to adsorb a molecule given that started at position y at time 0. It satisfies equation 360, which in this case reads as follows. Since the left boundary is adsorbing, the average time to adsorb a molecule is 0 if we start at the adsorbing boundary. So tau at time 0 is 0, while we have the reflective boundary condition at the other end. Integrating equation 475 and using these boundary conditions, we obtain that tau y is this quadratic function, which we have plotted as this blue line in figure 46d. There is also another line in this figure, this red dashed line, which corresponds to a slightly modified problem, where both boundaries are adsorbing. So we use boundary conditions tau at 0 and tau at L is equal to 0, instead of boundary conditions for 76, to obtain tau as this quadratic function of y. In summary, questions about the average time for adsorption are easy to answer by applying the theory which we learned in chapter 3. Another question of interest was formulated at the beginning of our lecture as: if we keep the density of molecules constant, far away from the boundary, what is the average rate of adsorption? That is, how many molecules are adsorbed per unit of time. We attempt to answer this question in 1D by solving the diffusion equation with the initial condition equal to a constant, and the limit of p as x goes to infinity equal to that constant. And we have a boundary condition at the left boundary x is equal to 0, p is equal to 0. As you can see here, the solution can be explicitly written in terms of the error function. We observe that the solution does not converge to a non-trivial steady-state profile. At any point x, the solution converges to 0 as time t goes to infinity. To understand this behavior, let us note how the one-dimensional model was obtained. We considered the full three-dimensional model, describing all three coordinates x, y, z by equations for 1, for 2, and for 3. In particular, our one-dimensional example of diffusion in the half-line interval 0 infinity can be viewed as diffusion in the three-dimensional half space. Even though the constant initial condition means that we have an infinite number of molecules available in the half space, we also have a very large adsorbing boundary, the infinite plane, which removes molecules at any point of the plane. In particular, the number of molecules adsorbed per unit of time is time dependent. To get better insights into above questions, we have to study three-dimensional problems. Then we will be able to consider diffusion to adsorbing surfaces which have a finite area. In section 4.4, we consider the adsorbing sphere of radius r0, and we find that the average number of molecules which are adsorbed per unit of time is equal to 4 pi d c infinity r0, where d is the diffusion constant and c infinity is the concentration of molecules far away from the adsorbing sphere. To derive such a result, we calculate the steady-state solution of the diffusion equation with the boundary conditions that the concentration far from the sphere is held constant, equal to C infinity, and the concentration on the surface of the adsorbing sphere is equal to 0. The solution of this partial differential equation is given by this formula. Since this solution is radially symmetric, the flux, that is, the average number of molecules which are adsorbed per unit area per unit time, is constant along the boundary r is equal r0. We can compute the boundary flux density as the diffusion constant times the derivative of c with respect to r at one representative boundary point. This gives us d times c infinity divided by r0. Consequently, the average number of molecules which are removed by the adsorbing sphere per unit time is given as a product of jr0 and the area of the sphere, 4 pi r0 square, so that we derive equation 471. Other three-dimensional problems are left to you as an exercise. In exercise 4.15, you are asked to consider a molecule diffusing in the following spherical shell, with the inner sphere of radius r1 and the outer sphere of radius r2. Both spheres are considered fully adsorbing, that is, whenever the molecule hits the inner or the outer sphere, it is adsorbed. Given the initial position of the molecule, you are asked to find the probability that it is eventually adsorbed by the inner sphere. To answer such a question, you can consider an auxiliary steady-state problem where molecules are released at distance r0 in such a way that the concentration at steady state is fixed at r is equal r0 to constant value c0. Then the steady-state concentration profile is obtained by solving the steady-state partial differential equation 467, that is, by solving Laplacian equation. In this spherical shell with boundary condition C at R1 is equal to 0 and C at R0 equal to C0. And in this spherical shell with boundary conditions C at R0 equal to C0 and C at R2 is equal to 0. Calculating the rate J1 of removal of molecules through the inner sphere and the rate J2 of removal of molecules through the outer sphere, the probability that the molecule is eventually adsorbed by the inner sphere is given by this ratio J1 divided by J1 plus J2. Once you calculate the resulting formula, you can also check that it makes sense. It is a probability, so it should be non-negative and less or equal to 1. If we start on the inner sphere, then R0 is equal to R1, and we obtain that the probability is equal to 1. If we start on the outer sphere, then R0 is equal to R2, and we obtain that this probability is equal to 0. So our result is consistent with our boundary conditions. A biological motivation for this example could be that R1 is the radius of the cell nucleus and R2 is the radius of the cell. And we are considering the behavior of molecules in the cytoplasm, which is the name of all of the material within a cell enclosed by the cell membrane, except for the cell nucleus. However, we can also use our result to answer exercise 4.13, which has slightly different motivation. In exercise 4.13, R1 is the radius of the entire cell. We consider an adsorbing sphere with radius R1 centered at the origin and assume that a molecule diffuses in the exterior of this sphere. So it can be a signaling molecule which can interact with the cell surface. And we are asking to find the probability that the molecule will be adsorbed by the sphere rather than wander away for good. To answer this question, you can pass to the limit R2 goes to infinity in your answer to exercise 4.15, and you will obtain that the probability that the molecule will be adsorbed by the sphere as R1 divided by R0. This probability is 1 if we start on the sphere, that is, if R0 is equal to R1. However, unlike in the one-dimensional case, this probability is less than 1 when R0 is greater than R1. So this is a fundamental difference between the one-dimensional and three-dimensional cases. In 1D, every molecule is captured, while in 3D, some molecules can escape and avoid being captured by the adsorbing sphere. Natural question is what happens in 2 dimensions. To get some insights into the two-dimensional case, you could solve exercises 4.13, 4.14 and 4.15 in 2 dimensions. That is, spheres will be replaced by circles and the spherical shell by an annulus. In 2D, our question can be formulated as follows. A molecule is diffusing in the annulus with the inner circle of radius R1 and the outer circle of radius R2. Both circles are fully adsorbing. That is, whenever the molecule hits the inner or the outer circle, it is adsorbed. Given the initial position of the molecule, you are asked to find the probability that it is eventually adsorbed by the inner circle. You can use a similar solution approach as in these exercises. So it will be about solving partial differential equations with the Laplace operator. Considering that a function phi in the n-dimensional space only depends on the distance r from the origin, the Laplace operator applied to phi can be given by this formula. In exercises 4.13, 4.14 and 4.15 in the lecture notes, we use this formula for n is equal 3, while for two-dimensional problems, you can use it with n is equal 2. This does not alter the approach to solving the two-dimensional problems. but it does change some answers. For example, the calculated probability in this case is equal to logarithm R2 minus logarithm R0 divided by logarithm R2 minus logarithm R1. If we pass to the limit R2 goes to infinity, we obtain 1 like in the one-dimensional case. That is, the probability that the molecule will be adsorbed by the adsorbing circle rather than wander away for good is equal to 1. We can better understand this conclusion if we consider a three-dimensional interpretation of this problem. The adsorbing circle is effectively equivalent to an adsorbing cylinder, which is infinitely long, so we have a much larger surface area adsorbing molecules than when we consider an adsorbing sphere in 3D, which has a finite area. In section 4.5, we consider reactive boundary conditions. To motivate this section, I have included here a picture from a biology textbook. It is a schematic diagram of a eukaryotic cell with various cellular compartments and membranes indicated. In our previous biological motivation, we mentioned that diffusion in a spherical shell could be viewed as diffusion in the cytoplasm, which is bounded by the outer membrane of the cell called the plasma membrane. This drawing is not to scale, and the width of the membranes is exaggerated because it comes from the part of the textbook discussing membrane proteins. Membranes, which encapsulate various cellular compartments, include a lot of proteins, called integral membrane proteins, which are illustrated here with a small segment of the plasma membrane. In particular, membranes are actively involved in many cellular processes, and if we want to model them, we cannot restrict ourselves to simpler reflective boundary conditions. We have already started to discuss boundaries, which are adsorbing molecules, in section 4.4. One of the studied models was the adsorbing sphere, where we obtained that the average number of molecules which are adsorbed per unit of time is equal to 4 pi d C infinity R0, where d is the diffusion constant and C infinity is the concentration of molecules far away from the adsorbing sphere. However, fully adsorbing boundary is a big simplification of a cell capturing molecules from its environment. It would be very dangerous for a cell to capture everything. It needs to have highly specialized receptors, which can distinguish between different molecules. We can refine this model by considering a sphere with adsorbing disks. In this drawing, only yellow parts of the surface are adsorbing disks, while we implement the reflective boundary conditions on the gray part of the surface. Then formula 471 is modified by dividing it by 1 plus 2 pi r0 divided by ks, where s is the diameter of these disks and k is the number of them on the surface. Considering that the cell radius is equal to 5 micrometers and its surface contains 25,000 receptors of the diameter 4 nanometers, which are uniformly distributed, we obtain that only a small fraction of the surface is covered by receptors, but they are still comparably effective in capturing molecules. Such a calculation shows that the cell surface can contain a lot of specialized receptors. From the point of view of the diffusing molecule, there are two different outputs each time it interacts with the surface. It is either adsorbed by the yellow disks or reflected back to the solution when it hits the gray area. This is also the way how we describe reactive boundary conditions in section 4.5. In this section, reactive boundary conditions are explained on one-dimensional models. Considering deterministic modeling in one dimension, it is described by one-dimensional diffusion equation for the density of molecules at point x and time t, denoted ρ in this section. Reactive boundary conditions are given in equation 481. Here d is the diffusion constant and sigma describes the reactivity of the boundary. That is, It is the rate constant of the boundary chemical reaction. There are two extreme choices of sigma which corresponds to the boundary conditions we have already discussed. If sigma is equal to 0, then we obtain reflective boundary conditions. If sigma is equal to infinity, then we can divide this equation by sigma to obtain the Dirichlet boundary condition that rho is equal to 0 on the boundary. This corresponds to fully adsorbing boundary, which we have discussed in the first half of this lecture. Considering stochastic modeling, the reactive boundary condition can be formulated in words as follows. Whenever a molecule hits the boundary, it is adsorbed with some probability, and otherwise it is reflected. So the stochastic boundary condition can be described by parameter p, which is the probability that the molecule is adsorbed rather than reflected. And our question is, what is the relation between p and sigma? If sigma is 0, then we have a reflective boundary, so p is also 0, and molecules are always reflected. We will not be surprised that p depends on sigma. If the rate constant sigma of the boundary reaction increases, then p will also increase. So p is an increasing function of sigma. However, p also depends on the stochastic model. Relations between p and sigma for four models of diffusion, which we introduced in sections 4.1, 4.2, and 4.3, are given as equations 486 till 489 on pages 128 till 130 of the lecture notes. To illustrate these conclusions, I have included here an example where I use the same value of P for two different models of diffusion. I simulate 16,000 molecules, which are initially uniformly distributed in the domain. I implement a reactive boundary condition on the left. Whenever a molecule hits the boundary, it is adsorbed with probability 5% and otherwise it is reflected. While I consider perfect reflection of molecules at the right boundary. The left figure was obtained using the compartment-based model of diffusion, while the right figure was obtained using the SDE-based model. I present the results at the same time for the same parameter values for the same initial condition, but these results are clearly not equivalent. The boundary condition at x is equal 0 removes more molecules on the right than on the left. To explain the observed difference, we can use figure 4.5a, which shows the value of the position of the diffusing molecule at times t and t plus delta t. If we use these two values, then we can conclude that the molecule did not interact with the boundary x is equal 0 during this time interval. On the other hand, if we decrease the time step, then the molecule could interact several times with the boundary. In particular, the number of collisions with the boundary depends on internal parameters of stochastic models of diffusion. For example, on time step delta t of the molecule-based diffusion model, or on compartment size h of the compartment-based approach. And this is indeed the case. If we want to simulate the reactive boundary with given reactivity sigma, we need to calculate probability p using these formulas. And we will obtain different values of p depending on which stochastic model we use. For my parameter values, I obtained that this probability is equal to 25% for the compartment-based model and 2.8% for the SDE-based model of diffusion. Using these values, we obtain comparable results, as it is shown in figure 4.7. This brings me to the end of lecture 15, where we discuss sections 4.4 and 4.5, covering boundary conditions for diffusion. Please read pages 117 to 124 of the lecture notes before watching the video of lecture 16. In our next lecture, we will discuss Chapter 6, covering stochastic reaction-diffusion modeling. As I have already explained in our introductory lecture 0, chapters of this textbook have been used as lecture notes for several different courses in the Mathematical Institute, University of Oxford, including basic undergraduate level and more advanced graduate level courses. Lectures on this YouTube channel are based on videos prepared for the most basic undergraduate level course, which does not cover more advanced topics from the lecture notes. The first chapter which is left out is Chapter 5, which will not be presented in our video lectures. Chapter 5 includes a discussion of a number of methods which can be used when your simulations become computationally intensive and your computer codes are very slow to run. It extends the covered material further towards the computations, and it can be of interest to some graduate students. While we have systematically covered the first 4 chapters in our videos, our remaining video lectures will only discuss some selected topics from chapters 6, 7, and 8. Thank you very much for listening to lecture 15 of our course on stochastic modeling of biological processes. Bye bye.